Hello, everybody. Welcome to the Lunchtime Discovery Series. Thanks for being here for another edition of our special program. I'm your host, Chris Smith, and I work here at the North Carolina Museum of Natural Sciences, where we broadcast this program to you every Wednesday at noon. An exciting opportunity to meet interesting people doing interesting things. This program is organized and coordinated by the folks at the North Carolina Office of Environmental Education within the Department of Environmental Quality, working together to bring you fascinating programs every single week. There's a great archive of Lunchtime Discovery Series programs here on the museum's YouTube channel. Make sure that you check out the Lunchtime Discovery Series playlist for all kinds of great programs. You can learn a lot by taking some time to scroll through and take in some of our past presentations. Really great stuff. Today, of course, I think we're going to have another great show. We have lots of great shows on this program. For the month of March, we've been celebrating Women's History Month. And in celebration and recognition, we've been hearing from some incredible women who are just brilliant people doing really cool work. It's been exciting to hear their stories, learn about their experiences and insights and expertise. And today, like I said, will be no different. As the program goes along, I'll remind everybody that the Lunchtime Discovery Series is interactive. That means you can jump into the program. I mean, you won't join us here on the call, but you can use the chat on YouTube or the comments on Facebook, wherever you happen to be watching. Leave your thoughts, your questions for today's guest speaker. After the presentation, I'll be looking to you, the audience, for your questions in order to pose to today's guest. So make sure that as we go along, you're typing those things up. That way it's all queued up and ready for me when we're ready for audience Q&A. Now, for today's special guest, they're actually, like, we're here on a video chat, but they're actually only like 3 floors above me inside the museum, I think, inside the genomics and microbiology lab. Dr. Heather Evans is a conservation geneticist with the North Carolina Wildlife Resources Commission, but has a really cool office here inside the museum. Dr. Evans, welcome.
Thank you. Thank you. I appreciate it. I'm excited to be able to speak, especially for the seminar on Women's History Month, and I'm included in this group of women doing really cool things in science. So let me start my presentation here.
Sounds good.
All right, everyone can see that?
Looks great.
Okay, then here we go.
Thank you.
All right, so yes, I'm Heather Evans. I am the conservation geneticist for the North Carolina Wildlife Resources Commission, and I'm going to refer to that as WRC from here on out, because that is a mouthful to say otherwise. But what I want to do today is talk a little bit about how we use genetics to conserve and sustain our state's fish and wildlife resources, which is the mandate that WRC has been given. So when I talk about things that we can do with conservation genetics, what I'm talking about are ways that we can use genetics to answer ecological questions. And that can be anything from looking at presence absence of species in surveys, to hybridization between species, or even if the animal that we're looking at is a new species or not. We can do things like looking at the genetic diversity of species, how many populations of a species there might be. We can also use DNA to look at questions of parentage or diet or health. We can even get some population estimates using genetics. So there's really a wide range of things that we can do. And I want to talk about a few of them today. But what I want to start with is more of the, well, how do we do that? So what I'm going to talk about is one of the methods that we use. Oh, I just realized my slide did not advance. So that was your conservation genetics toolbox that I just talked about. And so what I'm going to do now is tell you how we do those things. So one of the tools that I use a lot is something called a microsatellite marker. And I think I'm not still on the right slide. Chris, can you tell me what slide you're seeing?
Okay, let's see. It says microsatellite markers. They're 4 bullet points.
Okay.
And then 2 little text strands of, I'm going to guess, DNA.
Thank you. Sorry, my computer is not cooperating with me here. Okay, so that is the right one. So microsatellite markers, these are short segments of DNA that contain a repeating sequence. They generally occur in non-coding areas of the genome, and they're unstable because of that repeat, which means that every time it goes through DNA replication, it stands a chance of adding another repeat, which leads to a really high number of alleles. So a lot of times when people talk about alleles, they're talking about either like it's an A or it's a G, depending on the base pair. But with microsatellite markers, we're talking about alleles depending on how many of those repeats they have. So in this first allele here, what we see is a allele that has 284 base pair or 284 base pairs because it has 3 repeats. But a second allele could have an additional repeat, and that would add an additional 4 base pairs to that allele. So it would be 288 base pairs. So we measure those by fragment size. And then what we do with them is we can use multiple microsatellite markers and put together a unique genetic profile for species of interest. So in this example, this is an American shad. I'm going to talk a lot about shad today. And we use 12 markers when we're looking at shad. And so here you would see the 12 markers listed in this first column. The second column has the first allele. And again, this is measured in base pairs. So it's 301 base pairs. And the second allele for this first marker is 313 base pairs. So because of the variability that exists, each genetic profile is going to be unique to a specific individual. These are the same things that people use in forensics. So when you hear about a case and they're saying, oh, they're matching the suspect's DNA to the crime scene DNA, the way that they're doing that is with microsatellite markers. Now, how does this help us in wildlife genetics? So a couple of things that we can do. One is I'm actually going to start out talking about forensics and wildlife. At the commission, we do have a law enforcement side, and they are involved in cases sometimes, and sometimes I need to run genetics for them. So an example would be a case of illegal poaching. So we could have a private landowner that doesn't allow hunting, but comes out and finds a kill site on their land. There's some blood, maybe there's some animal remains. They can call our wildlife law enforcement and report that. Officers may come out and scout the area. If they find some hunters in the area with animals, they can take tissue samples from those animals. And then what we'll do is compare the animals to the kill site, just as in a human case. So here I'm showing an example. We have 4 markers, let's say, and we do genotyping at the kill site to make this unique genetic profile. Let's say there's 2 different animals that we want to test to see if they match. And if we compare individual one to the DNA at the kill site, we'll see that there are some similar alleles, but the majority of them don't match. So in marker 1, this 304 matches, but this 312 doesn't. Whereas if we compare DNA from individual 2 to the kill site, we'll see that 100% of those alleles match. And we can say, yes, this is the deer that was killed at the kill site, and law enforcement can proceed with pressing charges if needed. So that's one instance in which we use microsatellites. Now, there are other things that we use them for and more conservation related studies. So one of those are stockings and augmentations. The commission does a lot of stockings and augmentations for species that are in decline or even threatened or endangered. So this particular picture is a stocking project for American shad. What we do is we go out in the spring. So American shad are anadromous fish. That means that they are born in our rivers, then they swim out into the ocean. They spend most of their life in the ocean as adults, and then they come back into the rivers to spawn. So it's just like salmon on the West Coast, but on the East Coast, we have shad and some other species. So we go out in the spring when these fish are coming back in to spawn, we collect them and put them in these travel tanks here. This white tank is this travel tank, and then we'll take them to the hatchery where they put into these larger blue tanks. They're allowed to spawn in the blue tanks. We collect the eggs. We hatch them at the hatchery, and after about a week, we put them back into the river. So why are we doing this? Well, this shows a graph of commercial landings for American shad from 1950 to 2016. You can see that starting in the 70s, there's a really precipitous decline. What you're looking at here are these green bars. These are our American shad commercial landings. Start to decline in the 70s, and then in the early 2000s really drop off. So they have been at really historically low levels for the past 15 years. And so since 1998, the commission has been stocking American shad into the Roanoke River. That requires a lot of effort. Like, you know, see the crew that's involved here, the travel that's involved, the maintenance that's involved. And we want to make sure that these fish are surviving, because there's always a chance that you could take all these fry, put them into the river, and then they die immediately. So we want to know if our efforts are successful. So we need some way to tell if the fish that are in the river came from the hatchery or not, and if they're surviving. So traditionally, there were a couple of ways you could do this. You could use physical tags. This, right? Okay, here we go. You could use physical tags. This picture on top shows the a tag being inserted into a fish. You can see this is a small fish, but that's a lot bigger than this life stage that we're putting them into the river. The life stage that we're putting them into the river would not be amenable to a tag. So, and if we wait longer, it becomes more expensive because that's longer that we have to maintain them at the hatchery. You can also use chemical tags. Oxytetracycline, or OTC, is actually, it's an antibiotic, but you can put it into the water and it will stain a bone called an otolith, which is kind of this really fragile bone in the ear of the shad. And then you can then look later to see if it has an OTC mark. So this is a nice mark. It's a really like distinct, it stains well, but most marks aren't that clear. It can be very difficult to determine if what you're looking at is a real OTC mark or if it's, you know, just gradations in the bone structure. They do tend to wear off after a period of time. And so it's hard to get difficult counts. And one of the other issues with the chemical tags is that because it's in that ear bone, the otolith, you have to sacrifice the fish and be able to collect the bone to look at it. And since these fish are threatened, we don't want to be killing them. So we're going to use genetic tagging. So they'll take a fin clip from those fish. They'll send me this fin clip. It's about, you can see how big it is. So this doesn't hurt the fish to take it. It's like, you know, like if you were going to cut a fingernail, just the tip of the fin. I'll use about half of that to extract DNA. And then I'll use those microsatellite markers to make a unique genetic profile. We will do this for every brood fish that comes into the hatchery. So we have all the parents genotyped, and then we'll do the same thing for any fish of interest that we're looking at. And then just like when we were comparing microsatellite marker allele profiles for identity analysis, it's a similar kind of analysis, but looking for parents. So how it works is we have all the parent genotypes. We take our progeny of interest. We want to know if it came from the hatchery or not, and we'll compare those genotypes against each other. We have a computer program that does this for us. If you were doing it by hand, you could do it yourself, but we always have, you know, hundreds of fish to compare. So in this particular example, you have an offspring, it's 5201, and we ask, this is a maximum likelihood analysis. We want to know what are the chances that this parent, which is the best fit, is a true parent and it didn't match just by chance. So it tells us our best match is M406, and the chance, the likelihood that it matched not by chance is -27. So anytime you have a negative chance, that's not good. And we would not consider this then to be a match, because what it's telling us is that this parent matched, but so could 20 other fish match at the same level. Compare that to 5203, where it says, okay, the best match here is this parent 2031 with a likelihood of score of 19.4. For reference, if you have a LOD score of 3, that would be 1,000 to 1 odds that it didn't match by chance. So 19.4 is pretty good. And then it says the second best match was this individual, 2055, and it had a LOD score of 17.6, and then it puts them all together in a family unit. So what are the chances that these two parents produced this progeny 5203, and that has a trio then LOD score of 41.2. And so we would say, yes, this fish came from the hatchery. We do this for several hundred fish, 100, 300, and then can calculate the % hatchery contribution in the river. So for this example, 3 out of the 6 of our fish matched to hatchery brood fish. So that's our hatchery contribution is 50%. I'll show you some real data. So these are hatchery contributions for Roanoke American shad juveniles. The blue line is the hatchery contribution. The orange line is the number of fry stocked that year in the millions. So one thing that I want to point out is that the number of fry stock does not automatically correlate to your hatchery contribution. In 2012, we stocked similar amounts of fry into the river as in 2011 and 2013, but you can see we had very poor percent hatchery contribution that year. It was about 2.7%. So that's, again, just an indicator, which isn't surprising, that other things can impact your survivability of the fish. There are environmental factors, and some years are better than others. But we have had a couple of years with really high percent hatchery contribution. In 2011 and 2014, it was about 45%. And then what we're really interested though in is how many of those fish that are coming back into the river came from the hatchery. So I mentioned that they're born in the rivers, they go out into the ocean to live. They start coming back at around age 3 to spawn. And so we wanted to know, of those fish that are coming back to spawn, how many of those came from the hatchery? We started doing this genetic parentage-based tagging in 2010. And then because they don't come back in until around age 3, we didn't really expect to see any return on that until 2013. In 2012, though, we did get we got 2 fish that came in. They were young, but we did see some hatchery contribution that year. Now, we can only, in 2012, test back to 2010, because that's the first year we actually started taking those fin clips and doing genetic tagging. Anything that was stocked before then, we couldn't identify as hatchery contribution because we didn't have the genetic profiles. But as the years went on, we were able to add age classes. So in 2014, we see fish coming back into spawn that came from the 2010 cohort, as well as the 2011 cohort, and a couple from the 2012 cohort. And so our percent hatchery contribution has increased every year that we've been able to add an age class. In 2017, that was the first year that we felt confident that we had the full age class that was contributing to the spawn. You see a few fish from 2010 still contributing, and then 2011, 2012. The majority of the contribution came from 2013 age class fish, which would have made them age 4, which is typical, and then some from 2014 as well. And so the past 3 years, when we've been able to capture all age classes that are contributing to the spawn, we've seen an average of about 65% hatchery contribution. And that's pretty high. That's actually higher than we would like, because you have a trade-off in that you have a lot of fish here, but now they're all coming from the hatchery, which is a small genetic pool. So we have seen genetic diversity decrease over the years as our hatchery contribution has increased. So this was an instance where the genetic data helped make a management decision. In 2018, we decided we were going to halt stocking of the Roanoke River. We're going to take a pause and see if natural reproduction will pick up in the absence of hatchery stocking. We are continuing to monitor them. You can see we still monitored in 2019 to look at our hatchery contribution, and we look at our genetic diversity as well. But we're going to wait 3 years because it takes that long for the fish to come back into the river and start spawning. So we're not really expecting to see any results on this until 2022, when those age class fish that did not have stocking contributions come back, and we can see what the diversity looks like at that point. So that's one thing we can do, is we can look at parentage. Another thing that we can use microsatellites for are population estimates. So traditionally, when you want to look at population estimates, you would use a method called mark recapture, or some people call it capture mark recapture. And the basic idea is that if you take a population and you mark some number of animals in your population and then let them go back, they'll mix with all the other animals again. You'll come back on a second day, collect another subset of the population, and count how many of them are marked and have been recaptured. And that proportion that is marked should be proportional to your total population. So for example, in day 1, let's say we're looking at turtles, we go out, we get 10 turtles and we mark them. Then we send them back out. We come back on day 2, we collect another 10 turtles, but only 5 of them are marked. So to look at this proportion now, we would have, okay, we marked 10 the first day, we're solving for N, that's our total population. The second capture event, there were 5 that were marked out of a total 10 that we caught. We solve for N and you have a total population size of 20. So that's the main idea behind this. Now, the question is, how do you mark them? There's a few ways. So one thing is that biologists are pretty good about knowing what animals look like. So for example, if you were doing dolphins, they had different marks on their fins. If you can set up camera traps and you're pretty good about knowing, oh, that's Bob, we see him all the time. And then you'll see another cat come by and you're like, oh, that's a different cat. And you'll see how many times they cross that camera. But even though they're good at it, it's still really hard using a camera trap to be able to distinguish one animal from another. Another way is physical tags. So in this example, we have a deer who's been tagged. This deer has ear tags and a collar. You can definitely tell them apart now because there's an identifier. One thing that we are concerned about is the tags shouldn't affect their behavior. And so would wearing these big ear tags and collar affect their behavior? And there's some debate about this. And then here you could actually just write something on it, but it has to be something that won't wash off, which is harder than it sounds. So, and the other thing with the last 2 examples is that you have to be able to handle the animal then in order to get that marked. So there's another option, which of course is genetics, because that's what I do. And that is, we're going to use genetics to mark them. So with fish, you could take a fin clip. Here you see that's actually a pretty big fin clip. We don't need one that big, but you could go out one day, collect some fish, take a fin clip, go out a second day, take fin clips of the fish that you catch then, and then use the microsatellites to compare how many of these animals you've seen before. This first example, though, still involves handling the animal, and there are a couple of other options that you can do that don't involve handling the animal. So this picture in the middle here, it's hard to see, but this is barbed wire that these orange tags are hanging off of, and we wrap them around trees, and bears actually like to come and rub up against it. They'll use it to scratch themselves, and they leave us hair. So what you can do is you can come out every week and just collect the hair that has been left by the bears as they rub up against them. And then you don't have to handle the animal, which is great because it's a bear. Another option would be to use fecal samples. You can set transects and just walk in set lines and collect all the poop that you see. And then we can actually isolate DNA from the poop as well and make those unique genetic profiles. So a study would look something like this. This is a study by Ebert et al. That was published last year. And they are looking at 3 different forested areas in Germany. They wanted to get estimations of population for red deer. And so they set these transects. They were using fecal pellets. These lines are the transects. So they would walk along these lines, and anywhere you see a dot is a fecal pellet that they picked up and used for analysis. They genotyped them all, looked at how many of them were recaptures, and then using a more complicated formula than I showed you, but along the same lines, we're able to get population estimates for each of those 3 areas. And I'm not German, so I'm sorry that I'm going to butcher these names. But here, this HU is an area called Hunsrück, and their estimation for population size is 544 males, 560 females. So that's the other thing about genetics, is that you can not only get the unique genetic profiles with the population estimates, but we can put in a sex determining marker and tell you if it's male or female. So this way you get male-female ratios, which is great, because in the second area, which is Sunwald, we have 346 males and 600 females. So we have a 2-to-1 ratio of females to males, and really you want a 50-50 ratio. So now they know that their ratios are slightly off, and they can look at management options to see how they can even those ratios out. And then the third area, Falsterwald, they had 192 males and 295 females. This was their lowest density per square kilometer. And so they can now look and compare those areas, again, look at management options, or what might be going on in one forested area versus another that would cause the densities to be different like that. So you can get quite a bit of information using this genetic capture mark recapture method. I showed you a different study, not one that we've done at the commission, because we do have several of these currently in progress, but I didn't have good data to show. So we are currently doing these kinds of studies with our bear populations, both in the mountains and on the coast. We are also doing them with our elk populations in the mountains. For the bear, we're using hair traps. For the elk, we're doing fecal pellets. And we have also just started a study looking at white-tailed deer, and we're going to be comparing population sizes and densities in urban versus suburban versus rural areas in the Durham area. So that'll be pretty cool. All right. So that's genetic capture marker capture. That's another thing we can do with microsatellites. And now I'm going to switch methods a little bit and talk about a different kind of marker that we use, a different kind of technique, and that's called barcoding. So we call it barcoding because just like if you go to a store and an item has a barcode on it and you scan it, it will tell you exactly what it is that you're buying. There are certain genes within our genome that have evolved at the same rate as speciation. And so if you sequence them, you can know exactly what species it is. Most genes don't really do that. They're conserved because they serve specific functions and need to be conserved. But we have a handful of genes that do evolve in such a way that we can use them to barcode our species. So we sequence the gene, we can tell you what it is. These are names of some of them that I've listed here. I'm going to talk a lot about this one up top called CO1 or cytochrome oxidase 1. So just to show you an example, so let's say you have a tissue, you need to know what it is. You're not sure what it is. So we can sequence for CO1. Here you have cow sequence. This is elk. This was an unknown tissue sample, and this was deer. And so if we compare the sequence against each other, we can see that this unknown sample was elk, and we're able to ID it. So what other implications or methodologies might we want to use barcoding for? Well, let me tell you about surveys. So there's a lot of different surveys that we have to do within the commission. We need to know what all of our species populations are doing. Are they increasing? Are they decreasing? What are their habitat boundaries? So we spend a lot of time every year focusing on surveys for different species, but there's rotating schedules. And depending on how you're doing this, kind of it depends on the species that you're looking at. So for aquatic species, you can do electrofishing. So sorry, I forgot to move this again. There we go. You can do electrofishing. So this bottom picture is electrofishing on a boat. If it's smaller species, you can also do backpack electrofishing. They're in waders in the water, so they're safe. For mussels, you could do snorkeling. If you are looking at land animals and they have specific or distinct calls, you could actually do sound surveys where you set up recording equipment and then listen back to the tapes and pick up how many times you're hearing a sound, or if you're hearing the sound. So they do that with turkeys a lot, listening for gobbles. They can do it with certain species of frogs. And then the other one again is camera traps. There we go. These are really good pictures of, you can tell exactly what it is, but a lot of times it's night and you aren't able to ID it exactly. But the other option again is genetics. So we use what's called environmental DNA or eDNA, and you can take soil samples or water samples. You can take fecal samples. They're even starting to develop technology where you can take samples from the air. So the idea though is, let's say we take a water sample. There's a lot of DNA in there. We want to know what's in it. You can look for specific species of interest, or you can sequence everything that's in that water sample and get an idea of your biodiversity for a particular ecosystem. So if we were to take a water sample and then sequence it, we might get some frog DNA, might get turtle, fish, and it will tell you not just that you have fish in there, but it will tell you the species of fish. It will tell you have striped bass, you have American shad. So we can get really good information using environmental DNA, and again, without actually having to ever see the animal. We currently have an eDNA study going on Cape Fear. So the Cape Fear has a series of 3 lock and dams on the river system. The first one starts about 30 miles north of Wilmington. And then there's one in Elizabethtown, and there's one just south of Fayetteville. We talked before about American shad and how they come into the river systems to spawn. Well, because of these dams that are now located along the rivers, they only have access to that first 30 miles upstream of Wilmington. Then they hit Lock and Dam 1 and they can't get any farther. And we think that access to their historical habitat, their spawning grounds, might be one of the reasons that we see for the decline in their population numbers. This is also true for Atlantic sturgeon. So we have not only American shad, but Atlantic sturgeon that are coming into the Cape Fear. These are much bigger fish. And so let me show you. So we've tried to overcome some of these problems with the dams in later years. Let me back up a little bit, sorry. So I meant to explain how these lock systems work. So in previous years, a boat would come in here, would come into the lock, the gates would close, they would pump water into the channel and rise the water level, and then the boat could exit then on the upstream side. And as long as those lock and dams were working, the fish could also swim into the lock and dam and be transported to the other side. The problem is that we don't really transport things across up the Cape Fear like we used to, and these lock and dams haven't worked in years. And so now that these lock and dams aren't working, the fish also cannot get across these dams. So what can we do? Well, one of the things that they've done, this is Lock and Dam 1 now. The past couple of years, they've converted this from a dam to a weir. We basically put in a bunch of rocks and made it a gradual slope instead of a steep drop, and hope that the fish can get up that weir now and access above Lock and Dam 1. Now, we think maybe shad can get up that. We don't really think sturgeon can get up that. They're too big to navigate that weir system. So the U.S. Army Corps is going to do flood pulses. They just did one last week where they released water from Jordan Lake down the river. The idea is to flood just over the tops of the dams. They're not going to flood the surrounding system, the riverbanks, but they're going to flood it just over the tops of the dams so that the fish have a chance to get over those dams and access their spawning ground, which would be great. But we want to know if it's working and if the fish are actually taking advantage of that opportunity or not. So we're going to use eDNA to survey for those fish. This is my technician, Maddie. We're going out every week and collecting water samples at 11 different sites across, up and down the Cape Fear. We do above and below each of the lock and dams, and then a couple of sites in between as well. We'll sample in triplicate at each site. We put the water in these bags, and then we extract the DNA from the water, and we're looking specifically for sturgeon and shad. After we've extracted the DNA, we'll do a PCR to look for sturgeon DNA. And this is what it'll look like. So this is a PCR. You see these first, every line is a sample. And so these first 6 lines are actually positive controls. That helps us to know that the assay is working, and also helps us to quantify the amount of DNA that we think is in the system. And then the lines at the bottom, this line here is the threshold that we need to get above in order to be considered a positive hit. And the line down here then would be negative samples that never amplified. But these 3 lines here at the very end that get above that threshold are actually positive samples for Atlantic sturgeon. So far we have seen positive hits below Lock and Dam 1. We haven't seen anything above Lock and Dam 1 yet, but we did our first pulse last week, and we will continue to sample, and they're going to try and do another couple of pulses throughout the season. So we're hoping that we'll pick up some positive hits as the season wears on. All right. The other thing that we can use barcoding for is diet studies. So we talked about sturgeon a little bit. I showed you a picture of an adult sturgeon earlier. This is a picture of a juvenile sturgeon, much smaller. And we talked about habitat inhibition being one of the reasons we think they might not be rebounding. But another thing could be predation. Our fish and our river systems eating sturgeon, especially at early life stages. So we did a study last year. We examined 19 different species. We were especially interested in blue catfish, which is the picture in the top corner, because we have a lot of blue catfish and they are not native. So we wanted to know if there was a situation where we have a non-native fish now coming in and predating upon our native species. But we looked at 19 different species altogether. And of the 19, we found 8 that ate, our Atlantic sturgeon. Blue catfish did eat Atlantic sturgeon. About 6% of the blue catfish that we looked at ate Atlantic sturgeon. It's not huge numbers, but given the density of blue catfish could still be impactful. But we also found that 11.5% of common carp were eating Atlantic sturgeon. So actually more carp were eating sturgeon than the blue catfish. I will point out our highest percentage was striped bass at 12.5%, but we only had 8 samples. And so I don't trust that this percentage is real. But for the carp, we had 52 samples. The catfish, we had 134 samples. So that was those were pretty sufficient numbers. You can see also, like for largemouth bass, we had 72 fish that we looked at, and only 1.4% were eating Atlantic sturgeon. So largemouth bass don't seem to be a large contributor to sturgeon predation. Sorry. All right. So the last thing that I want to talk about is another barcoding gene called the control region. What's nice about the control region is that it's considered, we call it hypervariable. And it's not so much that there's a lot of variability in the base calls themselves, but there's actually variability in insertions and deletions. From species to species, there will be a lot of insertions, a lot of deletions, and different numbers, and that makes this particular gene a different size in every species. So this is nice, especially from a forensic standpoint. Because if you have an attack, so let's say we're having a rash of dogs being attacked by something, we're not sure what. When we swab the site on the dog, we'll pick up dog DNA as well as the predator DNA. So then when we try and sequence it, what happens is that you have the 2 sequences laying on top of each other, and it's messy and you can't actually get a good read for what it is. But because these are different sizes, we can use the control region and look at it that way. So in this example, you would see your dog DNA for the control region is going to be 393 base pairs. If it's a coyote that attacked your dog, we can tell that it's 388 base pairs. If it was a bear that attacked your dog, we could tell it was 425 base pairs. There would probably be other indications that it was a bear versus a coyote, but you get the idea where this allows us to look at multiple species at the same time without worrying about the contamination one sample to another. And so with that, I'll just thank my collaborators. Of course, NCWRC, I work with a lot of our staff on NCWRC who are fantastic. Because I'm the geneticist, I work with our species specialists often, so we can talk about the best ways that we can sample, the best methods that we can use to answer the questions that they have, both from a management or a conservation perspective. And then the North Carolina Museum of Natural Sciences, as Chris mentioned, I'm actually located here in the museum in the genomics lab. This is the lab that WRC uses to process all of our samples. The Federal Aid in Sport Fish Restoration Program funds several of my studies, including the American Shad parentage-based tagging study, as well as the diet study that we did. And then the Army Corps of Engineers, who is funding the eDNA study on Cape Fear. And that study is in collaboration with Clemson University and the Nature Conservancy. And with that, I will stop and take questions.
Excellent stuff. Everybody, let's give Heather a great big round of applause and say thanks. Really interesting stuff, cool stuff that I didn't realize was happening in the lab right here at the museum. Like all of this great animal DNA is just constantly flowing through our research labs. That's pretty exciting, I think.
Yeah, lots and lots of different species going through here, yeah.
And including like threatened and endangered species like the shad and the sturgeon. Like I've seen specimens of these here in the museum, and I've seen where we talk about shad or sturgeon in different educational facets of what the museum does. But and even though the museum itself isn't exactly engaged in this work, it's you and WRC. Like, it's pretty cool that WRC is so engaged in protecting species and using
Yeah, that's really the
really
mandate for
innovative things.
WRC is to conserve and sustain our wildlife resources. And so, you know, if a species is going to be state listed, that goes through WRC actually. And then anytime a species is listed, we need an action plan for how we're going to restore that species. And so all of that work is going on with WRC. Yeah, we have endangered mussels, we have endangered fish, we have endangered mammals, but all of that comes through WRC.
Well, we do have some questions for you in the chat, and I'll encourage everybody to keep typing them up and keep posting them. The first one that came in a little earlier in the talk was, how expensive is it to do the satellite marker DNA testing?
So the supplies themselves cost about $10 per sample to do the genotyping. If you're going to add in like time for salary and staff, it's about $50 a sample.
Okay. Not, I'm guessing that's not too bad. That doesn't sound too
terrible. It's not terrible.
And especially so much the infrastructure is built already here.
Yes. Yeah. One of the things that's really great, and the reason that WRC has this partnership with the museum, is that the museum has this great lab that's already here, where we have PCR machines, we have a robot that can help us process things in high throughput. We have a Sanger sequencer that we do the microsatellite analysis on, but also and the barcoding. And we also actually have a MySeq, which is the low end of the high throughput sequencing machines, but we do have one here, and we use that for things like the diet study, when we're going to sequence lots and lots and lots of things at the same time. So it's really well equipped here.
Excellent stuff. Okay, let's see. Does the amount of eDNA indicate density and abundance of the species?
So that is a question that a lot of people are working on right now. We would love to be able to get an eDNA. That particular graph that I showed was a special type of PCR called a qPCR. It's a quantitative PCR. Some people call it a real-time PCR, but it does allow you to quantify how much DNA was in your sample. The question is, can we then make any inferences about, does the quantity in the sample relate to the quantity in the environment? Maybe. Right now, people are working on this a lot. I think it's pretty good at doing relative abundance. So I expect that our amount of DNA in our samples will increase as the season progresses, because right now we're still at the very beginning of spawning season, and more fish are going to be coming in over the next month. So I think we can look at relative abundance that way. What people would love to be able to do is look at it and say, well, if we have this copy number of DNA, it equates to this many fish. And that's a harder thing. I'm not sure if we're going to be able to get there, especially when you're looking at water samples, because you have to take into account things like, well, where is the fish? Because there's flow rates. So the water is constantly moving, right? So I would expect that if you sample right on top of the fish, you're going to have more DNA than if you sample 100 yards downstream of the fish and that DNA has had time to disperse. So those are the things that make it tricky to use eDNA as a quantification tool.
Okay, that makes sense. That's sounds challenging.
Yeah, that was a challenging question. I think it's great for presence absence. If you want to know if your animal is in the system, it's great. It's trickier when you start talking about wanting to quantify it.
And along those lines, Cindy was asking, how useful might eDNA be for species discovery?
I think it could actually be pretty useful. We are going to do another study next summer for Roanoke log perch, which is endangered. And we'll be looking at some sites where they are known to be. So those are our positive controls. We're going to be looking at some sites where no one's ever seen them. So those are our negative sites. But we also are going to be looking at some sites where there's been some like dam removal, some habitat reconstruction, where they could be, but no one's looked yet. And so survey time to look for those endangered species, which are really hard to find, is a lot of work, but we're going to take some water samples and see if we can find them that way. But there are some previous papers have shown that eDNA has better detection than traditional sampling methods.
So if you're collecting samples through something like the barbed wire survey, where you're collecting hair, or you're looking for feces, and maybe it just didn't quite register with me, but that tells you if you find elk feces, you've got elk.
Yeah, so for sure. And we know we have elk, we can see them. In that particular study, what we're doing is trying to get a population. They're big, yeah. We're trying to get a population estimate. And so it's that idea of like, well, if we go out one day and let's genotype everything we caught in day 1, and then we go out in day 2 and we genotype everything, how many of those are recaptures? How many of those animals that we collected in day 2 did we also see in day 1? And then we can use that kind of statistical proportion method to come up with a population estimate.
Got you. Okay. Thank you.
Yeah.
Okay. Let's see. There are more here in the chat. I should take their questions. Okay. We've got a student who wants to know, what are some of the endangered mussels that you might work on?
So we have a project coming up that's starting in August. We're recruiting a master's student for it right now. We're going to be looking at dwarf wedge mussel, Atlantic pigtoe, tar river spiny mussel, and one that is dropping off my head right now. But those are a few of them that we're looking at. And what we're doing there is a couple of things. Like, one, we're looking at the population structure. So how many populations of these animals exist? Do they differ genetically from each other? And which of the populations have the most diversity? And that's important because the goal is to do breeding of these mussels and then augment or reintroduce. And we want to know, well, if we take mussels from this population, is it appropriate to stock them over here in this river system? Or do the mussels look different, so we need to keep them different? And also, which populations have the highest genetic diversity and therefore might have the best chance of survival if we put them in a new place? So we're going to be doing that. And then we're also going to be doing parentage-based tagging, like I talked about with the shad, but with the mussels, so that we know we're keeping track of what percentage of mussels in the river are now coming from the hatchery versus wild spawned.
So does the genetic work that you do supplant or replace things like field surveys or some of the other mark and recapture methods that you mentioned?
It doesn't.
Better than?
So it kind of depends. It depends on the system and the animal. In some places it's a supplement. In some places it's not adequate for what they need. And in some places it has replaced it. So for example, the parentage-based tagging, where they used to stain the otoliths, they don't do that anymore. We only do genetic parentage-based tagging because we decided that it was better, especially given the fact that we didn't have to kill the animals to get that hatchery contribution. And things like field surveys, we're doing, eDNA is still pretty new. And so we're doing both. It hasn't really been tested enough to make anybody feel like, yeah, we can just do eDNA surveys and never mind about traditional surveys. So we're doing both and comparing data, and we'll just make those decisions as we go along and collect more data. And it's probably going to be species specific because, eDNA in water, I think, is easier than eDNA with soil samples, because the water helps disperse the DNA for you versus eDNA for soil samples, you have to be on it. So that doesn't disperse as well. So it's going to, again, depend on the system. And we always do comparison, side-by-side comparisons for several years before we decide to drop one method. And sometimes we decide to do both, to keep both, because they're giving us different information.
Although I guess it sounds like the turnaround for getting the data and the information, maybe for making management decisions, can happen faster with genetics and the type of work you do?
It depends. So for things like the parentage-based tagging, those take about 4 to 5 months, but the wait is generally worth it considering the amount of data that you're getting back from it. Okay. For surveys, the eDNA probably takes a little bit longer, because if they're doing traditional surveys and they're out there snorkeling, they know that day if they saw it or not. The downside is they may not see it, and then they may think it's not there. And we may come in with water samples and be able to say, oh, no, it's there. You just couldn't find it. But it does take longer because we have to take it back to the lab and process it. So
different kinds of information to
yeah,
reinforce the questions then. So it
yeah.
really is coming at it from just several angles in order to get the best answers.
It is, yeah. So for that, the study on the Cape Fear, we're doing eDNA, but we are also doing electrofishing, and we are also doing telemetry, where there are physical tags in some fish, and we're going to pull all the data together, and that'll make a really nice study. We're going to have a really robust set of data to answer the management questions.
Excellent stuff. Okay, Judith wants to know if you're working with any plant species.
I am not working with any plant species. WRC actually had limitations on plant species, on being able to work with plant species. We have funds, these state funds, and I think we weren't allowed to work on plant species. There is a legislation currently working through Congress called RAWA, which is Restoring America's Wildlife Act. If that passes, then we will be allowed to use 5%, I think, of that money to work on plant species.
Oh, okay. I've heard of things like genetic barcoding for, say, tropical hardwoods. You know,
Yeah,
where they can track trees and things like that. So that's interesting.
yeah. The great thing about these genetics tools is that they can be applied to most any organism, because DNA is universal. So
All right, let's see here. A few minutes left in the program. Lisa wants to know how you got interested in conservation genetics.
that is a good question. I've always been interested in applied genetics. When I went to graduate school, my thinking was that I would do gene therapy. I would do medical genetics. But then when I graduated from grad school, I was easing back off. I had children, I had little babies. And so I was like, I'm just going to volunteer at the museum. Not unexpectedly, med labs don't usually take volunteers, but the museum did. And so I was able to come here and start volunteering and learn about applied conservation genetics, and just kind of took off from there. And so when I was ready to come back to work, I stayed in conservation genetics.
Oh, that's great. Yeah. You can't volunteer at a medical lab.
You can't, they won't let you. It's weird. They're missing out, really.
Obviously, there's great talent right here. Do you ever get samples, or receive samples and are just stumped on what's going on with the information that comes back? Like the sequences don't match up, the markers don't match up, and it just becomes that much more
generally,
of a
if you can get the sequence out, you have some idea about what's going on. The more of the stump comes in, like sometimes they will not sequence, or you can't get the DNA extraction out. So things like fecal samples can sometimes be difficult to get DNA out of. One, because there's inhibitors that don't like downstream reactions like sequencing. And so you have to run it through extra steps to try and get it clean and remove all those inhibitors. Or sometimes if you're working with a new species, you have a primer set that has always worked before, but when you try it on a new species, it won't amplify. And that's usually because the DNA sequence has changed just enough that those primers, when they bind, no longer bind, because your primer is a certain sequence and your animal that you're working with is a certain sequence, and they're supposed to match. So if they don't match, then it won't bind and it won't amplify. So usually it's, if I can get the DNA sequence that will give me some information, the trick is getting the DNA out.
So, okay, now, because this part kind of fascinates me, like how you actually get the DNA out. So do you have like a library of, I don't know, you said primers? You can go, okay,
Yeah.
well, book A didn't work, so let's try book B.
Kind of, yeah. So yeah, we have a lot of primers. We try to get like things that amplify really well across species. A lot of those barcoding primers. And we have different variations of them that we can try. With the microsats, those are developed per species. So whenever we're developing a microsatellite panel to look at a particular species of interest, it starts by taking that species and doing a high throughput sequencing, just basically fragmenting the DNA, shoving it on a machine, and it gives us back millions of reads. And then we data mine it for microsatellites and then design primers based on the sequence that we're getting out. So those are always designed per species. So we couldn't take like the microsatellite markers that we use on shad won't work on rabbit. But CO1 will work in rabbit and shad.
Okay, that makes sense.
Depends on, does it? It depends on the,
I think
depends
so.
on the gene.
Right. Yeah, sure. All right. Well, I'm checking here, and it looks like we're close to our time. So, Heather, thanks for being on the program today and sharing your work with us.
Yeah, thanks for having me.
Excellent stuff. Great to know what's going on with WRC all the time, because folks over at Wildlife Resources are just always doing really interesting stuff, like really cool genetics in the surveys, and just great. Well, maybe I just like working around animals, and I love that. But yeah, I've got great jobs that you get to do. That's in wildlife and wildlife DNA. So, hey, everybody, thanks for tuning in to another edition of our program. We will, of course, be back here again next Wednesday, so don't miss out. Go ahead and subscribe to the museum's YouTube channel. You can sign up to get email alerts about the Lunchtime Discovery Series 2 over at the North Carolina Office of Environmental Education's website, EEnorthcarolina.org. Or you can follow all of us on social media. The museum is at Natural Sciences on all platforms. The museum and the EE